personally salute all frontliners who have worked tirelessly and innovators who have yielded a solution to prevent and treat COVID-19 and other crisis situations, or an initiative which has contributed towards mitigating the impacts of coronavirus and providing relief to communities. I have observed abundant creativity and innovation emerging at the national, institutional, organizational, and individual levels for the past year.
Now is the ideal time for initiatives like this to be explored, not only encourage development of innovation, but to manage future emergencies and crises.
Yeah, I'm Dr. Ooi for MTE. I'm very appreciated. Actually, I very enjoy your video just now for the development of my country, Malaysia, for the advanced technology and biotechnology in recently. Thank you, Malaysia. And today we have a MTE Malaysia Technology Expo. So that's why we are here for the stem cell therapy web seminar for our future, not only just inside the video, even they are speaking in Malay, but they say not only for the Malaysia, actually for the whole world, just like us. Even we are in the online, we can talk together and promote together, not only in Taiwan, in Malaysia, but also in Australia. So I think today is a very, very good day for all your experts today. And we try to recruiting all of you together in one time, in one focus to Malaysia promote the advanced biotechnology, especially in the advanced cell treatment. So I'm very happy to have this opening remarks. By the way, I think I will introduce the next my professor, one of my teachers that I will very respect in Taiwan, Professor Hsing Jer.
As a chairman and chairwoman, and good friends from Malaysia and Taiwan. Good afternoon, everyone. I'm Dr. Shiing Jer Twu, the chairman of the Taiwan Development Center for Biotechnology, DCB. We know cell therapy is an innovative treatment that has been emergent in recent years, which utilize the somatic cells taken from patients or healthy donors to cure disease or regenerate tissue by infusing these cells back or transplanting to patients out of somatic cells differentiation, modification, and amplification. Currently, stem cell and immune cell are the two major therapeutic methods in global, and each has different development strategies. The difficulties of stem cell therapy products are cell bank establishment and maintenance, Therefore, the highly productive and strictly controlled manufacture procedures must take place to produce high-quality stem cell products. On the other hand, recently, immune cell therapy, infusing patients' own immune cells to attack tumor cells after gene modification and proliferation in vitro, has shown promising results. However, when we consider CAR-T, the most well-applied approach among immune cell therapy products, there are still inconsistencies in the outcome of CAR-T therapy, and the price of CAR-T therapy remains high due to the complexity of manufacture processes and the restriction of personal therapy and current stages. As a result, international pharmaceutical companies have started to develop allogeneic cell therapy. For example, the application of iPSC from healthy donors to establish hypoimmunogenic cell bank and generate universal off-the-shelf CAR-T for other allogeneic cell therapy products, such that the application of iPSC can reduce manufacturing costs and accelerate the application of cell therapy products. The cell therapy industry is the most active group in Taiwan biomedical industry recently. In Taiwan, more than 20 cell storage companies and 50 stem cell immune cell R&D companies. There are including 40 GTP labs and 4 GMP factories. Until February 2022, we have all 108 cell and gene therapy clinical trials, which including more than 30 companies, and have more than 55 trials, which is applying or ongoing status. With regulations of special medical techniques promulgated in September 2018, we have been development of the treatment advanced employing cell therapy. Until April 2022, we have more than 330 cases applied and 144 cell therapeutics approved by the Minister of Health and Welfare, and 46 hospitals joined forces with 25 cell therapy companies work together in cell therapeutics, and the result is quite promising. Due to the development of the characteristics of the cell therapy industry, the industry development models is diversified in stem cell and somatic cell research and development. There are such as unicocell, StemNet, MetaTech, etc. Many. And for the CAR-T and immune cells, we have a pair BMT, AV, life sciences, etc. Many. Number is increasing. A few companies shift their directions to developing new technology platform or new business model, such as focusing on the development of new technology and new cell types, or providing full services to a connected supply chain, that is CDMO, drug screening service, etc. The Development Center for Biotechnology, the DCB, now I'm a chairman, has been supporting biotechnology policy and promoting a number of R&D for new biotechnologies to tie in with the national policy for many years. DCB have developed iPS, derived neural progenitor cells, for treatment of sensor neural hearing loss, dopaminergic neuron for the treatment of Parkinson's disease, and the CAR-T therapy for solid tumor treatment, which are in the stage of validation and optimization. DCB aims to be the best partner in the biotechnology sector, not only for Taiwan, but also for the whole world. By integrating resources of the biotechnology research, DCB is able to accelerate the output value of biotechnology industry in Taiwan, as well as create new value for the biotechnological sector. Finally, I thank you for inviting for this conference and welcome everyone to Taiwan and welcome you everyone to cooperate with DCB in Taiwan. Thank you very much.
Thank you very much, Dr. Hsing Jer. I think yes, I think it's very appreciated your opening remark. Because of the times going on, we are supposed to begin the first speaker, Dr. Dai. Dr. Dai, he is the director of the Department of Surgery, now service in the Tri-Service General Hospital in Taiwan. Today, he want to talk with all of you about the cell therapy for chronic difficult work. Let's take your time, Dr. Dai.
Thank you, everyone. It's my great honor to join this great meeting. Right now, I would like to share my lecture to you. Okay, we can discuss after the lecture. Thank you very much. Ladies and gentlemen, it's my great pleasure to present the topic of cell therapy for chronic difficult wound. A chronic difficult wound is commonly defined as an unhealing wound sustained for more than 6 weeks. The important diseases include diabetic foot ulcer, pressure ulcer, venous ulcer, peripheral arterial occlusive disease, and radiation ulcers. Based on the Taiwan's National Health Insurance database, there are average 510,000 cases of chronic wounds a year. Moreover, it presented an annual increase in rates from 6 to 9%, indicating the serious medical needs for wound care. For developing and promoting regenerative medicine and cancer therapy, in 2018, Taiwan government legally opened up to 6 autologous cell therapies, including adipose-derived stem cells, bone marrow stem cells, collagen cell, fibroblasts, immune cells, and CD34 positive peripheral blood stem cells for clinical application. Here, I'm going to present the application of autologous keratinocytes and adipose stem cells for chronic wound therapy. Firstly, I would like to share with you the phase 1 clinical trials using autologous keratinocytes. The trial was conducted from 2012 to 2016, recruiting totally 10 cases of diabetic foot ulcer. The study group was designed to apply autologous keratinocytes with cell density of 5 × 10⁴ to 1 × 10⁵ cells per centimeter square. The control group applied with the aqua cell fiber dressings. In the protocol, the cell therapy will applied once a week until maximum 12 weeks or the day of complete wound healing. After that, 1 year following up period was applied to see whether there's the recurrence of the wound and the change of the wound size. As a result of this clinical trial, here is the first case of a control group. The initial wound size was 4.1 centimeter square. Aqua cell dressings was applied to treat the wound. The wound didn't heal actually after 12 weeks of therapy. In the end, complete wound healing was observed 1 month after in the following up period. There is no recurrence of a wound afterwards. For the second case of a control group, the baseline of wound size was 17.3 centimeter square. After the application of the aqua cell dressing, the wound size decreased gradually. However, it didn't heal after 12 weeks period as well. Finally, complete wound healing was observed at the 9 months time in the following up period. There is no recurrence after that. For the first case of cell therapy, the baseline of the wound size was 12.9 centimeter square, is quite large in the surface area. The cell density of 1 times 10 to the 5th cells per centimeter square was applied. The wound size decreased gradually after debridement and the cell therapy with increasing granulation rate for tissue formation. The wound size continuously decreased after cell therapy. Complete wound healing was finally observed after 9 times of cell therapy in this trial. There is no recurrence afterwards. For the second case of cell therapy, there is a diabetic foot ulcers over the lateral area of the right foot. The initial size was 0.9 centimeter square. Debridement and cell therapy were applied to this case. The wound size increased a bit after debridement and subsequently decreased after cell therapy. Finally, complete wound healing was achieved after 5 times of cell therapy. There is no recurrence in the following up period. For the third case of cell therapy, you can see it appears a significant inflammatory necrotic change over the wound bed. Adequate debridement was applied, following was the cell therapy. Continuous debridement was given due to the wound condition. The granulation tissue increased after debridement and the cell therapy. And after 4 times of cell therapy, you can see the wound bed condition improved significantly. However, the wound size was still in a large area. After 7 times of cell therapy, wound bed improved significantly with granulation tissue formation. After 10 times of cell therapy, a less necrotic tissue was observed. More granulation tissue was shown in the wound bed. Finally, incomplete wound healing was observed after 12 times of cell therapy. However, more granulation tissue formation was observed. A slightly decreased wound size was also observed in this case. In the following up period, complete wound healing was achieved at the 9 months time in the following up period. For the fourth case of cell therapy, the wound size was 1.6 centimeter square initially. After debridement, the wound size increased a bit and decreased subsequently after cell therapy. However, more keratinizing formation over the surrounding area was observed in this wound bed. And the wound size continuously decreased after cell therapy. However, frequent debridement was also needed to remove hyperkeratotic change of the wound bed margin. Finally, complete wound healing was achieved after 10 times of cell therapy. There is no recurrence in the following period time. In summary, for this clinical trial, totally 7 cases was recruited. 3 out of 4 cases in the cell therapy group healed within 2 months. In contrast, 2 out of 3 cases of control group healed after 3 to 4 months. In addition, there is no adverse effects was observed in this trial. As a result, the results show that the autologous keratinocyte cell therapy is safe and is potentially beneficial in chronic wound therapy. On the other hand, we would like to share with you the application of autologous adipose stem cells for chronic difficult wound treatment. For the first case, there is a gentleman with more than 70 years old, suffering from a diabetic foot ulcer over right heel for more than 6 months. Initially, we can see a necrotic tissue formation over right heel. After debridement and liposuction at the same time, we collected the fat tissue for adipose stem cells culture and processing. After 1.5 months for cell processing, we applied the cell therapy to the patient with the baseline wound size of 19.4 centimeters square. Adequate debridement was also applied to the wound bed. After 1 time of cell therapy, we can see there is some areas of significant granulation tissue formation. After 2 times of cell therapy, the granulation tissue increased significantly with slightly bleeding after debridement, and the wound size decreased slightly. After 3 times of cell therapy, more granulation tissue formation was observed. After 4 times and 6 times of cell therapy, decreased wound size was observed. And after 6 weeks of therapy, the wound size decreased to 12.8 centimeters square, with more than 80% granulation tissue formation. However, we can see a cleft of wound bed defects over the central part of the wound bed. Therefore, we performed the debridement and to close the defects. Continuous wound treatment was applied using the standard wound care modalities. And in the following up, we can see continuous wound healing was observed with decreased wound size and 100% granulation tissue formation. And after about 3 months of following up, wound size continues decreased with 100% granulation tissue formation over right here. So in summary for this case, we can say initially the wound bed size was 19 centimeters square. After 3 months cell therapy and wound care, the wound size decreased to 0.28 centimeters square with almost 100% granulation tissue formation. For the second case of the study, we can see is a lady with more than 80 years old who suffered from the diabetic foot, complicated with PLD, with chronic ulcers over right, first, second toes, and the first web space for more than 7 to 8 months. You can see in the screening stage, large surface areas of a wound with necrotic change over the toe tip was observed. After 1.5 months of cell processing, we applied the cell therapy to the wound. After a series of standard wound care, the baseline of the wound area was 4.3 centimeters square. And after adequate debridement, the wound size increased a bit. However, after 1 time of cell therapy, significant granulation tissue formation with contact bleeding was observed. After 2 times of cell therapy, the wound size started to decrease with more granulation tissue formation. After 3 times, wound continuously decreased with more granulation tissue formation. After 4 times of cell therapy, wound's condition improved significantly. After 5 times of cell therapy, wound size decreased to 4.9 centimeters square with 100% of granulation tissue formation. Finally, complete wound healing was achieved after 3 months cell therapy and wound care. There is no recurrence in the following up period. So in summary for this case, the initial wound size was 4.3 centimeters square. After 6 times of cell therapy and the standard wound care, the wound healed completely after 4 months therapy. In the third case, is a gentleman was more than 70 years old who suffered from diabetic foot and PLD with necrotizing fasciitis over right heel, complicated with osteoporosis and secondary fracture of a calcaneal bone. In the screening stage, you can see is a large surface area defect with the size of 48.9 centimeters square. After 1 time during the first time of cell therapy, the wound size was 78 centimeters square with more granulation tissue formation after a series of wound treatment. After debridement, the wound size increased a bit up to 82 centimeters square with more granulation tissue formation. Thus, after 1 time of cell therapy, increasing granulation rate was observed. After 2 times of cell therapy, more granulation tissue formation and decreased wound size were observed. In consideration of the wound bed improvement, we applied skin graft to the wound bed in combination with the cell therapy to the wound bed. After 3 times of cell therapy and the skin graft, wound size decreased significantly. However, there is a very deep hole over the heel area, and the depth was depth to the down to the bone area. Therefore, we did the adequate curettage and then applied artificial dermis pale neck in combination with the cell therapy to impact the hole. And the similar procedure was applied using the artificial dermis and the cells to apply to the deep wound. And the wound size over the outer area was decreased down to the 1.1 centimeter square with more granulation tissue formation. After 5 times of cell therapy, the depth of the hole is less depth compared to the prior weeks. The wound size change varies a bit to 2.9 per centimeter square. After 6 times of cell therapy, the defects shallowed and decreased in size, and the surface areas of skin defects varied, and up to about 5 centimeters square. And finally, the wound healed with good appearance, with residual rose surface areas over the heel. However, the defects complete healed was observed. In a summary for this case, for the initial wound size of 78 centimeters square, after a series of standard wound treatment, cell therapy, and the application of artificial dermis, the wound finally healed mostly after 4 months of treatment. So in conclusion, for the chronic difficult wound therapy, the policy for treating the chronic difficult wound will consider the pathophysiology of the wound, including systemic underlying disease, vascular disease, wound infection, necrotic, and significant depth of the wound. We can apply the drugs, standard wound care, and operation procedures, including the debridement, skin graft, to the wound. In addition, the cell therapy, according to our experience just shared with you, has a great beneficial to the chronic difficult wound therapies. Thank you so much for your listening.
Thank you, Dr. Tai. Actually, I enjoy your amazing your speech about the clinical cases of a difficult wound. I think we still have a few time to the audience can ask you the question directly. Any person online can ask the Dr. Tai the questions.
Thank you. Thank you so much for your listening. According to my experience, actually, I believe the cell therapy is wonderful options for chronic wound treatment. In the past, we have many developing in the district for wound care. There are many dressings, drugs, and any growth factors were being developed to promote wound healing. However, we need to remind ourselves that all the human beings started with just sterilizing germ cells. So that means we are all coming from stem cells in the very beginning. So therefore, the most important source for the life or for the wound healing are coming from actually the living cells itself. So after the clinical trials and clinical applications with autologous adult cells and also the stem cells, and I was convinced that the cells therapies, especially in the regenerative medicine fields, it plays a very, very important roles in the world. Just as the Professor Twu mentioned that, we actually has started to develop many some engineered cells or any other engineered materials to treat the disease or to heal the wound, and all this engineered technology are far advanced. I believe it will have a very great progress in the near future. At this moment, as a clinician, actually, we can have the cells in our hands very convenient. So I was convinced, and also I would like to convince all of you that cell therapy is for sure the very, very important weapon, the powerful weapons to treat the patient and to promote the healing, and also to prolong maybe in the future our lives. Thank you so much.
Dr. Dai, before you end it, you still have 2 minutes. Dr. Hwang want to ask you, I think in the message, did you use the immune pharmacy or treatment to treat the chronic wound healing beside the allogeneic cell therapy? Actually, this is also my question. I want to ask you whether you use any antibiotics or any combination, not only for the allogeneic cell therapy.
Thank you so much. It's a very, very important questions. Actually, based on my experience, treat the chronic difficult wounds in the past few years, I believe that actually as a clinician, we need to apply all the standard therapeutic modalities to treat the wound or to treat a disease. We cannot just to give up all the standard therapy and then just choose the new one. Okay. So while I am using the cell therapy to treat the chronic wound, I will firstly to apply the standard wound treatment technology. And for example, we need to control the infection. We need to remove the necrotic tissue, and we need to combine our standard or advanced therapeutic technology or surgical techniques to treat the wound. And however, I believe even we can do a very good surgery, even we can close the wound smoothly. However, the healing power comes from patient himself. So that means in the normal human beings, the healing seems to be the normal. So however, for the chronic wound coming in very ill patients, so the patients may not have the normal healing powers for themselves. So therefore, we need to apply some promoting healing modalities to treat the disease. That means we need somebody to help the patients. Just like in a war, you can see even in the battle, maybe both the enemy and our side are very tired. Once we got the friends coming from the sky, okay, and then they bring us the food, they bring us the weapon, and then brings us the people, the army, and then we can win the war in the end. So that's the similar concepts. If the patient cannot heal by themselves. We need to give them some helps. The cells is a very powerful weapons to help the patient to heal their wound. Thank you.
Thank you, Dr. Tai. I think we'll end the section here. To the second speaker, but Dr. Chung. Dr. Chung, now the next presentation will be in the video recorded. After that, and then in the end of the section, Dr. Chung will have a Q&A with us. But the speak will going through the video recording.
Hsing Jer. My topic is cerebral neurovascular reconstruction by stem cell sheet transplantation. Currently, I'm an attending neurologist and the director of Department of Neurology, Tri Service General Hospital in Taipei. And also, I'm an associate professor at the Graduate Institute of Medical Sciences of National Defense Medical Center in Taiwan. So it's my great pleasure to have this presentation at the Malaysia Technology Expo. So I would like to cover the following sections. So the first is unmet needs in neurology. So it's about brain tissue restoration as the medical unmet needs in neurology. Second, I would like to present about the scientific background about the stem cell biology and the tissue engineering. And then I would like to present how we manufacture the stem cell sheet and the preclinical study and the future clinical trials of stem cell sheet transplantation for the neurological disorders. Okay, so how can we restore the lost brain tissue and its function? I think there's still no satisfying answer to this question so far. For example, this is the computed tomography imaging of my patient who underwent the tumor removal of the meningioma at the right frontal lobe. And there's the permanent brain tissue loss after the surgery. And the neurological deficits all result from this tissue loss. So if we can restore the lost brain tissue, maybe we can improve his neurological function. So we have developed a new technology to brain reconstruction for permanent tissue loss after brain trauma. We established a rat model of surgical brain injury or brain trauma. We cut off the cortex of the rat brain, and later I will present how we restore the brain tissue by cell sheet transplantation. So here, not only the brain trauma, but also the stroke or dementia, Parkinson's disease, the degenerative disease, and like the patient I presented is the tumor removal. All these patients have the neuronal loss. So we have the brain atrophy in the elder age, and in this disease, like brain trauma or stroke, all the patients lost the neuronal tissues in the brain with no solution to restore this tissue and its function. So stroke is a global issue because 1 in 4 of us will have a stroke. It's just increased from 1 in 6 in the past 10 years. So more and more stroke patients, they lost the brain tissue due to the vascular events and no restoration of this tissue loss. So I'm currently the chairman of Taiwan Stroke Association. So we know stroke in Taiwan, there's some facts. For example, every year we have 40,000 new stroke patients, and more than 10,000 died of the disease, stroke. So the prevalence is about 400,000 stroke patients, and it's a huge burden in our family and the society. So the Taiwan market size about the stroke treatment and management maybe about a new start of 1 billion each year. So we developed the cell sheet and did the transplantation after we cut off the cortex of the rat brain. So 4 weeks later, we can see clearly the connection between the cell sheet and the host brain. So that's how we were honored or awarded with the National Innovation Award in 2018. So in the same year, we also got the Future Tech Breakthrough Award by the Ministry of Science and Technology in Taiwan. So in the United States, we know there's the market size for the traumatic brain injury. So it's about $2.9 billion each year. And it has increased, and they will have a bigger market size in the future. So like the traffic accident, even on the battlefield, traumatic brain injury is the serious issue for the civils and the soldiers. And about the cranial implants, so like the DBS for the Parkinson's disease, or maybe our cell sheet transplantation in the future. So the market size may be more than 100 million each year, and it's increasing in the future. So we would like to develop the new implant like the cell sheet. So it's the biomaterial or not the metal implants. But if we can restore the brain tissue and its function, so that will have a big breakthrough in this market. So that's the facts about the traumatic brain injury TBI in Taiwan. So more than 20,000 craniotomy each year, and the cost just only for the artificial memory, like the figure here. So that's the product of the Neurology Plus. So it's about $700 according to the Taiwan public health insurance the cost. So the market size in Taiwan is about 14 million each year. So it's all for TBI, but for the spinal cord injury, so this the first patient they presented last November in the Expo of the Biotechnology in Taiwan. So they did the mesenchymal stem cell transplantation by injection at the injury site of the spinal cord, and the cost of the price was $30,000, and we have the spinal cord injury patients more than 1,000 each year. So the market size may be $30 million each year. So in our hospital, we have a new unit called Cell Therapy Center from December 2018. So we have started the cell therapy for example, like the traumatic brain injury or spinal cord injury and the stroke. So this neurological disorder may be improved by this new technology in the cell-based therapies. And we, in Taiwan, we followed or take the Japan's experience as a reference. So in 2014, there's 2 acts about the cell-based therapy in Japan. So here I would like to present in the act. So the low risk is the stem cell with the homologous cells. But for the moderate risk is the non-homologous cells. And the high risk may be the embryonic stem cell. So if there's the homologous stem cell origin for the brain tissue restoration, the new stem cell is the only choice. So that's why some clinical trial shows the new stem cell as the cell source for the neurological disorder. So here we clearly can identify the neural stem cells by the characterization, if they can differentiate to neurons, oligodendrocytes, and the astrocytes. And nowadays we can read some reports using the iPSC as the cell source, but we have to see if there's the tumorigenesis, the risk of the tumors. So in the clinical trial, I will present is the PICES in the United Kingdom. They chose the new stem cells from the fetus brain. Also at this stage, the new stem cells can differentiate to all types of the neonatal cells. But still they cannot differentiate to like the blood cells or vascular cells. So that's how I have the innovations to combine new stem cells and endothelial cells to build up the cell sheet for the transplantation treatment. So in 2010, in the United States, they started the first in-human stem cell trial using the new stem cells for patients with chronic shock. So that's how they did. They did the surgery delivery by injection. So in their phase 1 clinical trial, they determined the dose of cells or the trajectory numbers. So they published their results in the journal Lancet in 2016. So that's the safety phase 1 clinical trial. And their phase 2B, they published the results in this journal, JNP, in 2019. So that's the PICES 2. And currently, there's the ongoing clinical trial, that's the PICES 3 study, that's the phase 2B in the United States and in Asia, several areas or regions. So even they check the MRI after the injection of the new stem cells, they couldn't find the newly generated brain tissue. So I mean, there's the injection of the new stem cells, but if we can combine not only the new stem cells, but other essentials like the cell or cellular components to make the cell sheet, maybe we will have a better results compared to PICES study. So that's the phase 2B clinical trial. So they focus on the motor function of their arms in these patients with the paralysis after stroke. So that's the chronic stroke. So 6 months after the vascular events in the brain. So they did the injection of the new stem cells in the brain to evaluate if there's improvement of the motor function, like the muscle power in their arms. So we are waiting for the results of this 2B. Okay, so that's the disease or the disorder of stroke. And ischemic stroke may accounts for 80% of all types of stroke. I mean, all types of strokes include ischemia or hemorrhagic stroke. And 1 in 4 of us will have a stroke. So it's a huge global health issue. So it's the pathologically, if there's occlusion of the blood vessel or circulation, so like the ischemic stroke, the damage of your from the vascular to the nervous tissue. So finally, the brain tissue or the neural tissue or injury results the deficits in the neurological function. So I mean, it's a vascular event, but the patient suffer from the neurological deficits. Okay. But in a physiologically situation, for example, the circulation in the brain could be regulated by the neuronal tissue or neural inputs. So there's the reciprocal relationship between the neural and the vascular compartments, not only in the physiological situation, but also the pathological disease like stroke. But how about the brain tissue restoration? So I studied how we have the adult neurogenesis in human brains. So in the specific neurogenic niches, there's the neural stem cells differentiating and proliferating through the whole life. So the neurogenic niches reside in the subimpedible zone about or surround the ventricle of the brain, and the other neurogenic niches reside in the subcranial zone in the hippocampus. That's the hippocampal region. So in these two, or in these neurogenic niches, we have the newly differentiated neurons in our adult brains every day. But when we have the shock or traumatic brain injury, these newly generated neurons cannot replace the damaged tissue. But we know not only the cell or the cell components in these neurogenic niche, there's for example, like extracellular matrix and other types of cells, like the vascular endothelial cells. They comprise the neurogenic niche together with the neural stem cells. So if I would like to restore the functional neurovascular unit, we may not only supply the neural stem cells, but also the endothelial cells or the extracellular matrix. So once we can combine these essential components to restore the brain tissue, like here there's the shock, it is the injection, like the transportation by the injection to the lesion sites, try to restore the brain tissue. And here there's the traumatic brain injury, so that's on the surface of the brain. They try to inject these cells. So what are the cellular and cellular components required for brain regeneration? So maybe not only the neural stem cells in the clinical trial in the PISIS study, not only the extracellular matrix, like some injection of the gels, but we can combine all these components to make the cell sheet to do the transplantation without injection. May have the better results. So about the tissue engineering, so here's the cellular components, and here are the acellular components like the biomaterials. And I studied the signals between these two components, and then we'll control this restoration process much better. So in Taiwan, we started the cell sheet transplantation for the knee joint, like the osteoarthritis of the knees, by transplantation of the cell sheet. And here's the cell sheet company in Japan. They did the cell sheet transplantation for several organs like the esophagus or heart or lung, but they did not or they couldn't do the cell sheet transplantation for nervous tissue. So here's no brain, the most important vital organ. But we established the first animal model to do the cell sheet transplantation to restore the brain tissue. I will present it later. So for the heart, there's already the clinical trial by manufacturing of a stem cell sheet, first in vitro in a dish, and this transplantation on the animal or human heart, or try to restore the heart tissue after myocardial infarction. So maybe after the cerebral infarction, if we devise or make an appropriate or the best stem cell sheet, you can also restore the damaged tissue, like here in the heart. So I published this in vitro model of a neurovascular unit or neurovascular environment in 2014. I established this novel in vitro model by combining the endothelial cells and the neural stem cells to make the neurovascular unit. So that's the discovery I published, that neural stem cells can induce the endothelial morphogenesis, when they combined cultured in appropriate conditions. Okay, so here we can see the confocal images of the neurovascular environment when we combine the neural stem cells and endothelial cells in the culture model. So we can see the green cells are the neural stem cells. So they may just on the vasculature structure of the composed of the endothelial cells. So the red cells are endothelial cells, and this is the endothelial morphogenesis. So that's the branching morphogenesis induced by neural stem cells. So in this article, I demonstrate that neural stem cells with different origins. So even for the neural stem cell transplantation, we try to choose the best or the most appropriate origin for the neural stem cells could facilitate the endothelial morphogenesis. So there's the branching morphogenesis of the endothelial cells after co-culture with the neural stem cells. And the different origins, the neural stem cells have the different or differential efficiencies in facilitating endothelial morphogenesis. So I started on the underlying mechanism of the differential efficiencies of the neural stem cells in facilitating endothelial morphogenesis. So some cells or some types of neural stem cells rely on the paracrine signaling. And some types of the neural stem cells rely on the juxtacrine signaling. So here's the in vitro cell culture model to show or to study the paracrine and the juxtacrine signaling. And we can choose the best neural stem cell in this model. So we published the results in 2016. So that's the first time we have the evidence to see how human neural stem cell induce endothelial morphogenesis. Okay. So here the blue one, the neural stem cells from the cortex rely on the juxtacrine signaling much more than the paracrine signaling. And here's the green one, so more efficient because they neural stem cell from the stratum, they can take advantage of both juxtacrine and paracrine signaling. So here we show the single cell sequencing of the cell sheet, and we know after the cells combined, culture together, the neural progenitor cells, how they change or the function shift when they cultured with the endothelial cells. So on the other side, we know how the endothelial cells change or the function shift after they cultured with the neural progenitor cells. So if we can elucidate the mechanisms or how they change after the co-culture, we know how they promote the brain tissue restoration after the cell sheet is transferred or transplanted into the brain. And we also characterize the extracellular matrix of the cell sheet. So for example, the fibronectin, vitronectin, or hyaluronic acid, they are all produced by these cells in the cell sheet. So not only the cellular components, but these cellular components like maybe the fibronectin or vitronectin, or normally they all contribute to the brain tissue restoration. So here we move to animal model to prove how the cell sheet could be transferred onto the surface of the rat brain after we cut off the cortex of the rat. And we did the functional assessment after surgery. So we can clearly identify the beta neuronal functions in the treatment group as the cell sheet transplantation group. So not only the body weight or the contralateral swing, but also there's the modified neurological stability score. So after the cell sheet transplantation therapy, the severity score became less than the control group on the day 28 after the surgery. So we can clearly identify the cell sheet in the treatment group connected with the red brain. So here's the gross view. So we can see the shallower lesion after the treatment by cell sheet transplantation compared to the control group with a deeper lesion on the brain surface. So here's the video to show the control group. So we cut off the cortex. So in 4 weeks, there's the permanent brain tissue loss. So that our patients with the traumatic brain injury, or in the first case I presented, there's the tumor removal. So they all lost their brain tissue and had the neurological deficits. So here the white cells are the astrocytes. So there's the glial scarring, so overlying the cortex without any newly generated brain tissue. So that's the control group. And here I would like to show the treatment group. So 4 weeks later, after we did the cell sheet transplantation, we clearly identify the multiple or many connections between the cell sheets and the host brain. So we can see there's the tubular-like structure. Maybe there's just the blood vessels in our cell sheet, and the white color is the other astrocytes. So not the scarring, but they just reorganized to form the functional neuroplastic tissue. And this one, we can see the green one are the astrocytes in the cell sheet, but the white one are the neurofilaments. So the neurofilaments can present like the neuronal networks. So in the cell sheet, we can see newly generated neuronal network. So maybe that's why there's the beta neural function in the treatment group. So we can see clearly the white neural filaments, like the neuronal network in our cell sheet. So not only for the brain or the central nervous system, but we did the cell sheet transplantation for the peripheral nervous system for the mice with the spinal nerve injury. So we can see the cell sheet transplantation to cover or to treat the injured gated spinal nerve, and the neurological function improved. So here's the IP of my research or my career. So the first two are the patents in Taiwan, and we got the US patent approved in 2020, and we have several ongoing applications. So I think there's the potential for these research results to be commercialized to help more patients. Now here's the US patent. We got the approved one 2 years ago. So there's some competitive advantage compared to other technology or patents, because we directly target the lesions on the cortex without the injection, because injection may make new injury. And we provide not only the cells, but also the extracellular matrix. All of these essentials contribute to brain tissue restoration. So here in Taipei, there's the BioMed Commercialization Center. So they make the YouTube online. So if anyone of you is interested in my research, maybe you can check it on the YouTube to see how we have the invention in this technology. So here's the Expo, also in Taipei. So not only in Malaysia, in Taiwan we have the Technology Expo. That's in 2018. So that's my presentation there. So finally, it's a summary to show the potential applications of technology of the stem cell sheath transplantation. So I think that's my career or my goal is the clinical trials to help patients with, for example, stroke or traumatic brain injury or like spinal nerve injury in the animal model and the future in the patients to help these patients to regain their muscle power to recover from the brain injury. And then we know there's a huge market size, so I will appreciate if there's any cooperation between you and our research group. And not only the clinical trials or animal trials, I think it's just the material and how they were made. So the materials for the cell culture, like the culture medium, is also the financial market. So the patent authorization is also very welcome if you are interested in my research. So here, that's my presentation. Yes, I think my presentation has run out of time, and I would like to join the panel discussion after all the speakers' sessions.
Okay, thank you, Dr. Chou. I think we are going to the third speaker, Dr. Hwang Xiaoming. He is the General Manager and CTO of U-Neuron BioMedical Inc. So today he want to give us the application of human amniotic fluid stem cell and their derivatives. Come on, welcome, Dr. Hwang.
Hello, everybody. It's my great honor to be here to talk about our company's focus on the amniotic fluid stem cell. And today my talk is the application of human amniotic fluid stem cell and their derivatives. Okay, that's my outline of my talk. First, just brief introduction. Then I will go through the details. Okay. And for the U-Neuron, we are a cell technology company and focus on the human amniotic fluids. And our mission is to provide this kind of cell therapy for provide innovative and effective cellular therapy for unmet medical needs. Okay. And as we know, the cell therapy has been recognized as the fourth and the final therapeutic pillar of the healthcare. And previously, and for the healthcare, the doctor has the three kinds of weapons to fight of the diseases. The first one is we call the pharmaceuticals, and the therapeutic product is the small molecular drugs. The core tech is use the chemistry, use the synthetic biochemistry to do, and the industry we call the pharmaceutical industry. And the second weapon we call the biologics. That's the macromolecular, or we call the protein drugs, or the monoclonal antibody. And the industry is the biotech. And the third weapon, okay, the doctor can prescribe is use the medical device. And the core tech is the physics and engineering. And the industry is the medical device industry. And still, there's a lots of the unmet medical needs. And the people thought maybe the cell to develop the cell therapy. And the core tech is, of course, is the cell and the tissue engineering. And the developer, the cell therapy industry can fit the unmet field. Okay, and that's the we call it the three pillars. That's not actually the health care. They can only talk about this called sick care. When people get ill, then doctor prescribe the pill and to kill the germs or the pathogens. But we know in the regenerative medicine, regenerative field, people get ill not because of the infection, they are because of their cell or tissue malfunction. The cells did not work well. In this case, you need to inject the right functional cell, and people can get well. Then that's the true health care. Okay, and this is the Deloitte 2020 report. Is collected the global cell and gene therapy clinical trial. Okay, they count total is more than 8,000, and in this all cases you can see the green one. This more than 3/4. That's over the 75%. It in the CGT belongs to the stem cells, and the 1/4 is the CAR T, the immune cells and gene editing, etc. And you can see the more than 75% is belong to the stem cell clinical trial. That means a lot. And why we choose using the amniotic fluid stem cell? As the first, I will like to explain what is the amniocentesis. Amniocentesis is a routine prenatal diagnosis procedure when fetal cells from the amniotic fluid are obtained during the 16 to 20 weeks of pregnancy. That's we call a second trimester. And the doctor use a long needle to under the sonar. Okay, ultrasound guided. Then we'll draw around the 20 ml amniotic fluid. And in this amniotic fluid, there's a lot of the fetal cells. And in these procedures, they will check the fetal cells chromosome. We call the chromosome karyotyping analysis. And in this picture, you can see the right picture. That's the yellow, yellowish, the liquid in the syringe. That's the amniotic fluid. That's no blood cells. Okay, but again, this only the fetal cells, not from the mother. So we call this the prenatal diagnosis procedures. And for the amniotic fluid stem cell history, back to the 2007, from the JAMA, there's a review article. They say the stem cells obtained from amniotic fluid. They recognize the amniotic fluid contains the stem cells. And at the same year, the Time magazine chose the finding of the stem cells from amniotic fluid as the top 10 medical breakthroughs. And so that's the reason, finding the stem cell from amniotic fluid, and the functionality of these cells is between embryonic and adult stem cells. And this cell is scalable and safe and low immunogenic. So we propose that maybe it has a great promising in therapeutic field. Okay, and that's the generation of the mesenchymal stem cells. Okay, the first generation is from the bone marrow. Okay, people found the MSC from the bone marrow, and then people found the adipose tissues. Okay, like the Dr. Dai mentions, they use in the chronic wound healing cure. They use the adipose derived mesenchymal stem cells. And then the third generation is they found from the cord or the placenta also contains a lot of the mesenchymal stem cells, and those are very young. They can use for the allogeneic, and there's the zero age and the rich source. But in the medical uses, they are treated as a birth medical waste. But if there's from the natural birth, there's a worry about the microbial contamination. And how about is there any younger cell source? We found that yes, the fourth generation of the stem cells can get from the amniotic fluid, and that's also for the allogeneic, and also for there's the youngest fetal cells, and we take the amniotic fluid from the pregnancy of 16 to 20 weeks, and that's the routine prenatal procedure, no ethical issues like the embryonic stem cells. So in this trend, you can see the MSC generation is from much younger and no ethical source, and also move to the non-hematopoietic tissues from the bone marrow to the amniotic fluid. And we are the first group to isolate and culture and characterize the stem cells from the amniotic. We published in 2004 in Human Reproduction. We call it the isolation of human multipotent mesenchymal stem cell from second trimester amniotic fluid using a novel two-stage culture protocol. And also there's that issue of the cover story is use our paper. Also, we applied a patent and quickly has got the patent to culture these amniotic fluid stem cells. And these cells will perfectly fit the International Society for Cell and Gene Therapy. We call the ISCT. They publish a minimal criteria guideline for the MSC, and there's 3 criteria for MSC. If you want to call your cells are MSC, the first is these are attached cells. Yes, these are AF amniotic fluid stem cell is attached cell, not belong to the hematopoietic stem cell. That's a blood cell. That's a suspension. These cells are attached. And the second is the surface marker is totally the same as the bone marrow MSC. We can see there's the 29 positive, 44 positive, 105 CD73 positive, HLA class 1 positive, but not the CD31, CD34, CD45, and the HLA class 2. And the third is that this cell needs to be differentiated into the osteocytes, chondrocytes, and the adipocytes. That's the 3 lineage belong to the mesenchyme. So from we call it the AF MSC. That's from the amniotic fluid derived MSC. That's the perfect fit, the ISCT criteria. Besides that, we found the amniotic fluid MSC contains some of the embryonic stem cell marker. We call the pluripotent markers, including the SSA EA4 and the NALOG. So because these cells are very early, the fetal cells. Beyond that, we can check these cells. We call the youngest cells. Then we can check the telomere length. That's the chromosome, the end of chromosome. I can use the pointer. Like this one. We call this is the telomere length. We also call the biological clock. The younger the cells, or the longer the telomere, once they divided, the shorter of the telomere length. Okay, when people get older, the telomere length will be shorter. Okay, and that's the left side. You can see the amniotic fluid has the longest telomere length compared to the cord blood MSC or the bone marrow MSC. Okay. And we also published more we developed a microfluidic method to isolate this kind of MSC. And we also found, okay, this amniotic stem cells expressed both the mesenchymal stem cell and the neuron stem cells. That means the AFSC has the same marker of the NSC, neural stem cells. So this kind of cell may be fit. The Dr. Zhou Zhongxin has the last talk about mentioned the source of the NSC. Okay, and we also checked this AFSC can be benefit for the peripheral nerve regeneration and also in the Parkinson diseases. And this cell have the potential for the angiogenesis, something like that. Okay. And people also use the amniotic fluid stem cells to do the stroke. Okay. They found that these cells can proliferate and attenuate behavior deficit in ischemia stroke rates. They inject the amniotic stem cells into the brain, okay, the cerebral parts, and they found that the cell can engraft in the damaged parts, okay, and differentiate into the neuron cells or the secrete the neurotrophic factors to help the recovery of the ischemia stroke rates. Okay, and also the people found the amniotic stem cell can apply in the kidney diseases. They can prevent or slow down the renal fibrosis, or we call the renal interstitial fibrosis, and the loss of the papers. Okay, use different kinds of the kidney models. They can show the benefit of amniotic fluid stem cell in this field. Okay, and also from they can apply the amniotic fluid stem cells in the lung diseases. Okay, well, they can inhibit the progression of the pulmonary fibrosis, and that's we can find that also people use the amniotic fluid cells to do the COVID-19 patients to prevent the lung fibrosis. Okay, when we checked the amniotic stem cells can apply into the stroke, Parkinsonian diseases, or peripheral nerve injury, and some people use the amniotic fluid stem cells apply in the heart diseases, the PAOD, or the diabetes, kidneys, liver, cartilage, and the lung diseases. There's a lot of the disease models. You can see the AFSC also applied in this kind of field. Okay, and I summarize the advantage of using the amniotic fluid stem cells. The functionality, we call this the pluripotency. Not like the ES embryonic stem cell, we call the pluripotency. And the adult stem cell, we call this multipotency. So the fetus amniotic stem cells is between the ES and the adult stem cells. So in our field, we call this potency, we call it as the pluripotency. It's less than pluripotency, but better than the multipotency. You can see. And the source is no ethical issue. And these cells, no tumor formation. Okay, it's verse to the pluripotent stem cells. Okay, like the ES or IPS, they will form the teratoma. But for the AFSC, no one reports the FSC will form the tumors. Okay, and these cells grow fast, and the scalability, and because they are very young and HLA class 2 negative, they are low immunogenicity. So that's perfect for the allogeneic stem cell source. Okay, and back to the mechanism of the stem cells, the more and more paper and more people, including us, found maybe that's the paracrine effect. We call it the cell-freed, okay, AFSC derivatives. We can collect the conditioned medium or purify it as a secretome or the exosome. And this conditioned medium contains lots of the growth factor and the function like this. And besides growth factor, there's a anti-inflammatory cytokines, also the glycoprotein and ECM. And of course, there are some of the exosome. So now more and more people, probably they more acceptable to use the cell-free products. Then there's a more focus on the secretome or the exosome. Okay. For example, in this, you can find the lots of the paper. They called the using the amniotic fluid stem cell extracellular vesicle as a paracrine tool endowed for the regeneration potential. Okay, there's AF exosomes. Okay, from the amniotic stem cells, their therapeutic perspective, and some of the example, for example, in the ischemia, the stroke model, ischemia reperfusion model, they used the AFSC secretome. They showed a perfect neuroprotective effects, and also in the other for reduce the Alzheimer's diseases, something like that. Okay, and for this AF exosome, or we call it the extracellular vesicle, the production is probably is the same as the flow chart. Okay, the first we have the IRB, and to approve this kind of experiment or this kind of to accept the amniotic fluid, then expand the amniotic stem cells and collect the conditioned medium. Harvest this conditioned medium to do the centrifuge tangial filtration, then size sieve purification, then do some formulation. Then there will be your products. We call it the AFSC EV products to do the disease model, and then to apply the clinical trial. Okay, and that's some of the picture of our facility. We have the GTP compliant cell processing unit. Okay, and we have the liquid nitrogen storage area, and this is the we call the CPU cell processing unit. Okay, the room. Okay, and our collaborators, a lot of the medical hospital in Taiwan. Okay, let's say we call this our company. We call it the U-Neuron. We want to do using the amniotic fluid stem cell for the neurological diseases. Thank you. That's my talk.
Okay, thank you, Dr. Hwang. Yeah,
Okay.
really enjoy your talk. I'm respiratory physician. I certainly have a question to you.
Sure.
Did you any experience or any paper published about the post-COVID-19 lung fibrosis or organ damage from your fetus stem cells?
No, we didn't apply the COVID-19 patients the trial. No, not yet. Okay.
Oh, not yet.
We tried
because yes, but because during your presentation, I think the fetus can recovery many organs, and because of post-COVID-19 damage many organs. That's why I'm very interested that whether you have experiences for this. Any persons or any doctors here have experiences for the post-COVID-19 cases?
Can I ask a question?
Yes, you go on.
Very congratulations, Dr. Hwang. You present that amino-free stem cell, right? You
Yes.
of course stem cell, right?
Yes.
And you mentioned many times is there's low immunity. Also, that you mean that this can be used allogeneic, right?
Yes.
So
Yes,
did you
like the MSC, like
Yes.
the other MSC. Yes.
So did you pull so many different resources of amino-free together and try to harvest more amino-free stem cell from different pregnant mothers together and try to incubate together to see if something you can mix together, right?
No. We found a different donor set the different amniotic fluid stem cell bank.
Yes.
That's only yes. We have the last different donors. Then okay, and then we will choose the first one to do the clinical trial. Yes.
Why you choose one? Because this is no immunity. You can use allogeneic, right? So
Yes.
in theory, they can pull together and become a kind of together. I just want to know if you have done this to see that if you mix together, what happens to these cells? They fight each other or cooperate each other or just make you more efficient because you can pull together like culture easier. Is that?
Oh, okay. For the regulatory view of point, okay, that we are not allowed for a mix of the different donors. That's good scientific the studies the issues. But I guess if you put 2 or more donors AFSC together, they still will compete each other. Because the different donor has a different growth rate, a little bit the differences. The one is the grow faster, the one is the slower. Then when you expand it, the grow faster will dominate or the one. So this
But
will.
anyway, you don't know yet. But
Well, I
you
don't know.
want to become a kind of commercialize and produce a dot of that F stem cell. Then later you must prove this is workable, or you can incubate a culture individual one. But finally, you must put together. You can use only one. I'm able to treat one person. Patient.
No. Okay, Dr. Twu, because we just get 5 ml amniotic fluid, then we can expand it. Then we do a master cell bank. Then we use the master cell bank to produce the working cell bank. Then one donor can at least 1,000 dosage usage, just like the other bone marrow MSC you can expand it.
Later, maybe you must choose which AF free stem cell is the best for use.
Yes.
Because you can reproduce a dot, right?
Yes.
So you must choose which one is better for commercialize.
Yes.
Theoretically, can apply to any other people, right?
Yes.
Okay.
Yes.
Very interesting discussion both of you. I think we have very good future. We are going. Time is limited. We are going to the fourth speaker. Dr. Yang. Dr. Yang
Okay.
from Taiwan Bio Therapeutics Corporation. Dr. Yang, it's your time, please.
Hello, everyone. Can you see my screen now?
Yes, I can see it.
Okay, perfect. So today I want to share with you a little bit about how to translate genetically engineered cell therapy from bench to bedside. Oops, sorry. So many of the speakers have has introduced you about different kinds of cell therapies. But I think nowadays, because of the success of CAR T, the focus of cell therapy development has gradually shifted from non-engineered cell to the engineered cell. And as we all know that CAR T is very popular, there are similar kind of other immune cell like CAR NK or even CAR macrophages. But genetically engineered MSC or other cell types are still kind of like trailing behind of those immune CAR cells. So today I want to talk a little bit about the general principle to consider the development to be considered when you develop a CAR T therapy or genetically engineered MSC therapies. So I would like to start with some general principles of making genetically engineered cells, or afterwards we'll call it GE cells. So I think the whole story all began when Emily Whitehead was treated with the very familiar anti-CD 1920 CAR T, to treat the leukemia, and become very, very highly successful. And since then, I think she's gone more than 10 years free of cancer. And compare this development with the previous immunotherapy, non-GE immunotherapy like TIL, tumor infiltrating T cells or other NK cells, one can easily conclude that genetically engineering can basically empower the T cell or other cell types to perform better job when we try to combat disease. And this trend is also very apparent in the regulatory field. So here is the recently approved ATMP from United States and EU. So as you can see here, most of them are cell-based gene therapies, and those are actually genetically engineered cell like CAR T or genetically engineered CD34 cells. Only very few of them are non-engineered cell therapy, like at the bottom here we have mesenchymal stem cell and matrix-associated chondrocytes. So you can see that genetically engineered cell are very, very prominent and dominant. They are dominating the field. And however, after the success of CAR T, there are a lot of manufacturing issues started to become more apparent. For example, we see that the first CAR T approved by US FDA, the Kymriah from Novartis, they have concerns of those cells being out of spec, and we still face a lot of the logistic bottleneck for CAR T production and scale up, and that's mainly because CAR T, despite its power to treat cancer, it's an autologous therapy, right? So you invariably will have patient-to-patient variabilities in the starting materials, and you won't be able to scale it up because for every patient you need to take their blood, isolate T cell, make them CAR T. So those problems making CAR T, despite its clinical success, very hard for many patients to access. And this is why there are a lot of companies trying to improve the CAR T production manufacturing process. And I think this is very critical because in Taiwan Bio, we are not only a cell therapy new drug developer, we also has a large PXGMP cell therapy facility, and we do provide CDMO services for our clients. And one common theme we encounter when we serve our client is that manufacturing upgrade or optimization is a very, very difficult process in the cell therapy field. Most of companies don't know how to do it. A lot of regulatory agencies have huge issues to decide whether to product after the manufacturing optimization, whether they are the same product or the different product. So I think if we want to streamline the development process of genetically engineered cell, we need to start at a very early stage to think about how we're gonna optimize our manufacturing process to make our cell sort of like more industrialized. The manufacturing process become more industrialized. So we frequently talk to different regulatory agencies, and there are some common theme they mentioned, like comparability studies are inevitable because throughout the development process you will have change of the manufacturing side, like you find a new CDMO service provider, you will try to scale up your process, you will change from FBS based manufacturing process to more like a chemically defined medium, or you will start to introduce automation to your manufacturing process. And after each change, each optimization, you will need to perform comparability studies. And our experience is that regulatory agencies would like you to start those comparability studies earlier, right? So they would rather you to start the comparability studies in phase 1 and phase 2, try to optimize your process, and in phase 3 they want you to have a settled process that will not be changed. And after phase 3, it's very hard to further improve your process, and a lot of time additional clinical trials will be required by the regulatory agency if you want to improve your manufacturing process in or after phase 3. And that's mainly because cell therapy products, unlike large molecule, antibody, or small molecule, they are very complex, right? So a cell can do many different jobs at the same time, and the mechanism of actions are more complicated for cell therapies compared to the traditional small and large molecules. So even with our best effort to perform comparability studies, we will inevitably face some issues with, like, if you change a parameter of a process, it can change the property of the cell and potentially affecting its therapeutic effect. So to perform comparability studies, FDA have some guidelines, but I think the main focus here I want to mention is that it needs to be a scientific process, and it needs to be statistically sound, right? So you still need to follow the logic of science to design your comparability study. And the general scheme to do these kind of comparability studies, that you first need to have a very clear mechanism of action. So you need to know what your cell therapy does in the body to treat disease. And then you can develop a target product profile, basically characterize your product, what's the features, what kind of disease you want to treat, what's the administration route, etc. And from the mechanism of action and the description of your product, then you can define what are the most important attributes of your product. So we call those critical quality attributes. So those are the attributes that will affect your product quality, the safety, and the efficacy of your products. And once you have these defined attributes, you then can define what kind of process parameters will affect your quality attributes. For example, if you try to change the condition, the culture medium of your cell, because the ingredient there will be different, it may affect cell proliferation, it may affect the growth factor cell secrete to the surroundings, and those will affect the critical quality attribute if your mechanism of action is depends on your cell to release a lot of different growth factor and cytokines to achieve the therapeutic effect. So once you can define all those things, basically you have characterized your product, you have defined your process. This will make your development process much easier to be optimized at a later stage. And I think this is a very important take-home message here, that to streamline the from bench to bedside process, you really need to have this scientific approach to characterize your product as early as possible and define the quality of your products. And I also listed here at the bottom a term called KPP, so key process parameters. That's because, as I mentioned, cells are very complicated living biopharmaceuticals, or regenerative medicines. They do so many things. Some of them may not be quantifiable, and some of the quantifiable parameters we may not be aware of while we're developing the process. So, at a later stage of the clinical development, we would like to have a defined process that will not be changed, like I mentioned earlier, like in phase 3. And those we will define. After we define those process, we will have those key process parameters also being determined, so we can ensure the quality of our product are very consistent. So I just want to mention briefly in this slide that the mechanism of action of your product will affect the target product profile of your product, and that will drastically affects how you design your clinical trial and in your process. For example, these are the same product, same CAR-T product. They want to use it for different purposes. So one is use a bridge therapy to hematopoietic stem cell transplantation, and the other is to use as a standalone therapy to treat cancer. And you can see that the regulatory agency will have very different requirements for the clinical trial endpoint. For example, if it's a bridge study, the clinical outcome measures will not be as strict as when the CAR-T therapy being used as a standalone therapies. So for a standalone therapy, you may need to measure overall survival or event-free survival. However, if you are using it as a bridge study, you can measure MRD status or the subsequent rate of HSTC, etc. So that makes the trial much easier to do. And all those will affect how you design your process later. So I think, again, I just want to mention that this is a very scientific-based approach. You need to really have a very clear MOA and how to treat your disease to make it successful. So once we understand the overall process of develop product, then we can go back to check, what's the difference between genetically engineered and non-engineered cell. So I think the main difference for the engineered and non-engineered cell is that for a lot of non-engineered cell, like the mesenchymal stromal cell we always talk about, generally you need to understand the cell's intrinsic properties, like what kind of cytokine and the growth factor is secreted, and you need to observe whether they have the immune modulatory ability. However, for a genetically engineered cell, you can have a desired MOA, right? So you can have a very clear understanding of a disease pathogenesis, and you design a transgene to achieve a certain process. For example, we know that mesenchymal stromal cell is very angiogenic. They can secrete a lot of angiogenic factors, and to increase their angiogenesis, you can put a transgene in there to increase the expression of those angiogenic factors. And so you have a very clear design major MOA. And of course, you still have to characterize other characteristic of your cell product, because even with a transgene, the cell will still produce a lot of other cytokines, chemokines. They will have their homing ability, immunomodulatory ability. Those probably won't be affected. So you still need to characterize your cell. However, the major mechanism of action can be very, very clear. And after that, you can characterize your product and design, what's your clinical trial, what's the disease you want to treat, so you can have a more rationalized cell therapy design, from the administration route and to the dosage, etc. So, for example, if you have a genetically engineered mesenchymal stromal cell that can overexpress a certain angiogenic factors, you can measure the concentration of those angiogenic factors they secrete to the surroundings. And then you will know, what's the lowest amount, the minimum amount you need to achieve for a single cell in the surrounding to achieve the therapeutic effects. And then you will know whether you can achieve the desired angiogenesis in human body. And once you know the TPP, then you can go to the CQA. And for the CQA, I think this is a drawback of using a genetically engineered cell. Although it's much more powerful, its design and its CQA definition and its processing become much more complicated. So you need to understand not only the intrinsic properties of your genetically engineered cell, but also the property of transgene. And let's take CAR-T as an example again, right? So we know CAR-T expresses CAR, and the CAR has the active domain which recognizes a particular tumor-associated antigen, and they also have this intracellular domain which can stimulate T cell signaling like CD28 for 1BB, etc. And from the regulatory point of view, each functional domain, or let's say the biologically relevant functional domain, you will need to characterize whether they does the exact thing you wanted to do inside the patient's body. So the overall definition process become much more difficult. And the CQA become more difficult, become more complex. The CPP also become more complex because you need to start thinking your product from the vector design, the plasmid you want to transduce or put in your cells to the formulation. And the overall approach to define CQA is sort of like a feedback approach, right? So first you need to have a candidate CQA. You develop a qualified assay to detect those CQA, like the angiogenic factors we've been talking about. You need to define a specification. For example, like what's the concentration, minimal concentration you need to achieve per 1,000 cells, and you try to correlate those CQA with your product quality, test that in a clinical trial. So if all goes well, you have a very good CQA. However, if your angiogenic factors, for example, turn out to be not correlated with the therapeutic effects, then you probably need to go back to this feedback loop again and to identify another CQA. And because the complexity of cell therapies, we generally recommend that a matrix of CQAs, so not depending on only one factors, but multiple factors of your cell therapy product, will be highly desirable throughout the development process. So after expanding the overall principle, the general principle of developing a genetically engineered cell, I want to give you a specific example using a genetically engineered MSC to treat critical limb ischemia. So a little bit background about critical limb ischemia. It's a lower limb. It's a major cause of lower limb amputation. It has very high prevalence in the elderly. And it's actually the most severe form of another disease called PAD, so peripheral artery disease. So basically, the pathogenesis is that you have a diseased artery, so you will have a decreased blood flow, so your blood cannot carry enough oxygen and nutrition to the lower end of your body, or lower extremities. So you will have resting pain, you will start develop some severe unhealed wound, and generally you will lead to, as I mentioned before, amputation. And critical limb ischemia, or CLI, is one of the major cause of amputation in type 2 diabetic mellitus patients. So in Taiwan, we have very high prevalence of type 2 DM. So this is a huge, very severe disease. And previous study have identified that angiogenesis may be a very important therapeutic strategies to treat CLI. So you can see that there are a lot of different vectors being tested, especially gene therapy being tested to treat CLI. So the idea is that, although the artery is diseased, we try to create microcirculation to circumvent those diseased artery to provide oxygen and the nutritions. And gene therapy for CLI has some problems. They have low transaction efficiency, and the MOI is very hard to control. So basically, how many copies go into a cell is very hard to control. And study also identified that single factor is less effective than the multiple factor. For example, overexpressing FGF and VEGF at the same time is more effective than HGF alone. So people start to think about using angiogenic cell therapy to treat CLI. And the study shows some promises because, as I mentioned, cell can do multiple things. They can secrete multiple growth factor, multiple angiogenic factors. However, the expression of those growth factor will generally be affected by environment. And although a lot of trials show some preliminary success, show some promises, there's no improvement in the most relevant clinical endpoints such as amputation rate. So one problem we identified throughout the study of CLI is that we think for non-engineered mesenchymal stromal cell, they do not secrete enough angiogenic factors, and their expression are generally affected by the environment. So we really want to have an overexpression system to overexpress the most important angiogenic factor that is VEGF. But before we go to VEGF, we actually do a panel of screening. So we have a panel, a list of candidate panel we want to overexpress in our cells. So that include BFGF, PDGF, TGF beta, etc. But what we want to do is that we don't want to disrupt the intrinsic property of a cell too much. So we check their proliferation rate, we check their function on the mesenchymal stromal cell we tested. So in this case, we're using bone marrow derived mesenchymal stromal cell. So we test their triangle differentiation to see whether they will be affected. And interestingly, we can see that VEGF doesn't really affect the cell proliferation rate, and it don't really affect triangle differentiation. And we try to keep our MOI low in the mesenchymal stromal cell, so we can have less disruption in our cells. And I think this is a very important topic. Like you choose a clinically proven safe vector, for example, like third generation lentiviral vector, and that has been previously used in the clinical trial. And we test their MOI, and you can as you can see here, even with MOI 1. So basically, on average, 1 copy per cell, we can have almost like 7-fold of increase in VEGF. At the same time, the cell can also produce other angiogenic factors, cytokines, and they have a similar immune modulatory effects. So we believe that this product will be quite successful. So we tested in a mouse model. So basically, here you can see that we create the artificial CI model, and the blue shows that the blue part shows that there's less or decreased blood flow, and once we treat them with our GE cell, you can see the blood flow start to getting back to improve, basically. And it's important to not only compare our cell with control, but also compare our cell with non-genetically modified cell. And as you can see that the blood flow increased significantly after 45 days compared to the non-engineered cell. So with all those data, we are now trying to push these to the clinical trial to get an IND approval from the US FDA. And as I show in this case, that we start with the pathology of this disease, CLI, and we start by identifying what's the mechanism of action. So basically, we want to develop vasculature around the disease area. So as you can imagine, like those cells will be injected directly around the disease artery, and so that's basically our product characteristic. And we developed that we developed a comprehensive TPP to describe this product, how we're gonna treat those disease, and then we define the critical quality attribute of these cells. So of course they need to secrete a lot of VEGF. We also check whether they express other growth factors. We check their immunomodulatory abilities, and then we start to define their process. So the original process, when we started this project in collaboration with a UC Davis group, they were using FBS to produce those cells, but the whole process is already in a fully closed automation system. And what we do is basically we try to optimize this process further by switching from FBS-based system to chemically defined culture medium system, and try to increase the batch size, and try to introduce cells bank system to this genetically engineered cell. So by doing this genetically engineered MSC, we can actually make allogenic genetically modified cell possible. And because it's allogenic, it can circumvent a lot of issues CAR-T face, like for example, like the patient-to-patient variability I mentioned before, and the expensive QC process per batch, and difficulty to scale up, etc. So hopefully we will be able to share more with you about this genetically engineered cell. And as you can imagine, intergenesis is such a crucial process. Yeah, I think I'm finished on my presentation.
Okay, thank you, Dr. Yang. Because of the time is overrun the time. Dr. Wei Thomas directly, and after that we can have a Q&A in each other. Please take your time, Dr. Wei Thomas from Australia.
I'd like to thank you for the kind invitation to talk today at the Malaysian Technology Expo. And can you hear me now?
Yes, can hear you clearly.
Yes.
Yes.
And the chair, Dr. Wu, fellow speakers and guests.
We can.
Are you able to hear me now?
Go ahead, sir. Go ahead. Yeah.
Today I'd like to talk about the adipose cellular fraction, indications and mechanisms. But first I'd like to talk a little bit about the regulatory frameworks and cell therapies. There's, we've heard a lot of talks this afternoon, which have been very good talks on the different regenerative medicine therapies and different cell types. And as you can see from this particular publication, there's extremely a lot of advances happening with numerous international studies and research protocols on the use of stem cells, the tissue engineering, cell reprogramming, and genetic therapy. And though cell therapy policies appear to vary significantly throughout the world, with overlapping jurisdictions, with international organisations, nations, and even states. Differences seem to be occurring even with the two largest regulatory bodies, the USA FDA and the European, on the way we treat cell therapies. So from what I can observe, each country, it's difficult to navigate the regulation of this complex and ever-changing field of new medicine. So each country will implement their own different regenerative medicine policies based on their own safety, economic and health reasons, and also with an eye on the huge potential market for therapeutic applications on a global scale for regenerative medicine. With Australian regulations, our regulatory body looked at the stem cell field and very much decided on a risk-based approach rather than a strict regulatory approach as such used by the US FDA. And they decided that the TGA, the Therapeutic Goods Administration, do regulate human cells and tissues, but they don't regulate medical practice. And they looked at autologous cell therapies as being under medical practice. And they had three main criteria for us to do cell therapies, in that it was collected underneath a doctor and manufactured within the hospital on the site and not advertised to consumers. This allowed medical practice in Australia to continue to use standard therapies, autologous therapies, such as skin grafts, vascular conduits and bone grafts, etc., without having regulatory issues, and that now included such autologous treatments as stromal vascular fraction, where we focused our energies and technology. So I'd like to now move to talk about adipose cellular fractions. There are a number of various sites around the body that we can obtain adult regenerative cells, such as the MSCs, the mesenchymal stem cells, such as the bone marrow, the adipose tissue, umbilical cord, synovium, and the fetal synovium fluid, as we just heard. But it appears that although these cells are very similar, each of these sites give various characteristics to their cells, such as the bone marrow have a very good differentiation potential. The adipose tissue has got strong immunosuppressive effects, and the umbilical cord it's got great self-renewal. The synovium fluid here has got low immunogenicity. And as other talks have previously indicated, there's various criteria for determining MSCs, such as differentiation into adipocytes, chondrocytes and osteoblasts, and they may also differentiate into the endoderm or the ectoderm. The focus of our technology has been on the separation of adipose tissue into the regenerative cell mix, which is termed stromal vascular fraction, or in our case, because of the technology that we've developed, we call it StroMed. And we decided on adipose tissue because of its ease of abundance, and it's the largest microvascular tissue in the body, and it's able to provide a very extensive bioactive extracellular matrix. Over here is a DAPI stain that we've done on an adipose globule, and you can see here these round cells here are fat cells compared here to the vascular tissue, the blood vessels running through the fat cells here, and this is where we obtain our stromal vascular cells. And in amongst these blood vessels, there's the fibroblasts, the stromal cells or mesenchymal stem cells, endothelial progenitor cells, pericytes, macrophages, T cells, and erythrocytes. Within the SVF cell populations, what we've generally found that the stromal cells, the mesenchymal stem cells, generally range between 10 to 30%, and the pericytes around 5 to 20%. But the breakup into this population tends to rely on two things: a method of separation, and also on individual patient variation, which may be due to such things as obesity or smoker or general fitness. The stromal cells have generally got, these cells range in size. Generally, they say the stromal cells are generally around 10 to 25 microns in size. And once they reach culture, they may go up to about 200 microns in culture. This is probably why you see a difference in the literature about problems using these culture cells passing through the lungs, which you don't see with the more the cells directly obtained from SVF, and also the stickiness of those cells in culture. The smaller cells are generally defined about 7 to 8 microns, but my belief is some of these cells we see are actually smaller than that in size. As with all things in the scientific literature, concepts keep changing and continue to develop about cell therapy and the cells. One of the more current concepts is by Kaplan here as to where the MSCs are located, and that the pre-MSCs, PMSCs as he terms them, may be sitting on the outside of the blood vessels, with pericytes there, along with other niche cells. The theory is current that perhaps when the blood vessels are broken, they release these perivascular cells that differentiate into the MSCs. And these may be the pericytes which differentiate into the MSCs. So the pericytes are also a very important cell type there. And we've also noted in our therapies that we see the pericytes playing a very important role. As other talks have indicated, there's very good regenerative modes for these stem cells, such as immunomodulation, anti-apoptotic effects, angiogenesis, and support of the growth of local differentiation of local stem and progenitor cells and chemoattraction. Many of these cell types within SVF are themselves going on to have further studies on their individual cell types, into clinical studies, into stroke, autoimmunity, heart disease, pulmonary disease. Our thoughts with our technology is we believe it's actually conducive to the action of the MSCs and the other cells, that there's placing these all together as there's lots of crosstalk happening between these cells, And I think it's been said in the literature there's over 200 growth factors released, and this crosstalk is happening all the time, helping to in the regenerative modes of these cells. So our technology sources the SVF from autologous adipose tissue, basically because fat is 100,000 times more the number of stromal cells than bone marrow, and we can do this as a bedside process. In Australia, we performed the first SVF treatment back in 2009 and started international work about 2012. And we advanced to a clean mechanical system, ultrasonication, to produce the SVF, which we term StroMed because it is slightly different from other methods, and we believe, a greater cell type, number of cells, and slightly different cell types. So our patented technology, we believe, offers significant advantages over the competing technologies, such as it's clean, there's no risk of disease transfer, it's autologous, no risk of rejection or allergenic reaction, easy to harvest, there's a high number of cells harvested. You can see here the cell pellet, same day, same clinic, and it's safe as ultrasonic cavitation was used for decades and can be low cost. So in I suppose very quickly, our cell therapy, we perform a mini liposuction, a small amount of adipose is obtained because we only need a very small amount of adipose because of the high number of cells that we obtain. We process it within about approximately 30 minutes. And then we do a flow cytometer cell count where we look at the viability and the dose number to ascertain the exact cell number that we're going to inject for the particular disease. It's very important to understand that cell number which is being injected into the site. And then we can deliver the StroMed cell preparation in about 5 minutes. And you can use this preparation as IV, depending on the indication, because it is a single cell. It's not I suppose, a complex of fat in that mix. We have focused our attention on the use of SVF in orthopedics, and it has been our leading indicator, the osteoarthritis, even though there are plenty of opportunities to focus our attention on different diseases, as I think all cell therapies are focusing on a lot of diseases, chronic diseases. And I think each stem cell will have different uses, and which stem cell will be the best use for which disease we will eventually work out. So we've been focusing on knee osteoarthritis as it's been one of our key indicators, as it's very much suited prior to surgical intervention at grade 4. Current treatment options is where you have NSAIDs, physical therapy, cortisone injections, steroids, and lubricants. But they really do not do much to improve or delay the disease. So we see cell therapy as being a very key intervention prior to any surgical intervention. And the hope is to delay the disease or regenerate the tissue. With knee osteoarthritis and SVF, we see there's 4 potential roles that the cell therapy can play. The first being in pain modulation, mobility of the patient, slowing of degeneration, and repairing the cartilage. Any one of these would be a win, but But if you can do all four, and that's been shown in our data and others around the world in the literature to occur with cell therapy. So I'll just quickly go through the evidence in the literature, what's out there. So this is why we think SVF is really starting to become a good treatment for osteoarthritis. There's 41 currently referred publications on this, and this is close to 3,000 patients in these studies. There's 3 level 1 studies, 4 level 2, 5 level 3, and approximately 30 level 4 studies since 2021, or prior to 2021. And all 41 of those studies, except one, showed clinical improvements in pain and mobility, and those that measured cartilage demonstrated cartilage improvement, and a number of scientific validation tests were undertaken, such as WOMAC, COOS, VAS, ROMs, and MRIs. So when you look at the literature in summary, it appears that grade 3 improvement ends up being in grade 4. It's the best outcome, and multiple studies have shown this. The use of platelet-rich plasma, which is used to help with SVF, it seems that it improves by about 10% approximately in the literature, and knee extensions have been improved, and one injection appears sufficient. And now clinical improvement has persisted to greater than 5 years in the literature, and our studies have gone out now to about 7 or 8 years and continue to show persistent improvement with our patients. And these studies have showed MRIs, T2 mapping, MOCARTs, and arthroscopy have verified the improvement or maintenance of the cartilage. There have been numerous safety reviews, 5 years on multiple joint injections, intravenous infusions, and no serious adverse events or evidence of tumour formation have occurred. There's been minor adverse events reported, such as knee pain, swelling, infection, joint injections. There has been recently some publications where people have used SVF and put it into the eye, and there has been some serious adverse events occurring. So I think that was very, very silly, I suppose, study to undertake with patients. With our data, one particular cohort of 80 patients, we observed after 12 months and approximately an 80% improvement, and that's measured by the WOMAC study into the knee joint, and this was using both intraarticular and intravenous injection of the cells. Another study which we performed on approximately 240 patients, we followed the patients for 3 years post-SVF, and this was a partial intention to treat analysis. And as you heard, intention to treat analysis is one of the more harder statistical analyses to do in following patient cohorts. And after 3 years, we ended up approximately about 64-65% improvement in the patients. But to put that in context, when you look at a total knee replacement using WOMAC studies, which we used here, you only get approximately a 60% improvement in the literature on total knee replacement. So I think we're pretty comparable to TKR. In this particular cohort, our median age of patients was 66 years, and they range from 20 to 91 years, with our median weight of 84 kilos and a range of 40 to 148 kilos. We did see individual patient responses where some patients improved rapidly over the few weeks starting. Some patients responded slowly and improved it over time. And some patients, a quick response then went backwards, and we think that was generally due to overuse of the joint. We've also seen with our studies, we see cartilage loss here. Week 7, an improvement in the cartilage, and by week 16, we see a good improvement in the cartilage. But that's not to say you're going to see cartilage improvement in all patients. If they've got focal lesions, we see some good cartilage. But if it's bone on bone, what we've seen 3 years out is from a biopsy, good tissue growth happening, but also wear and tear at the same time. So there's a lot of weight and pressure on those knees. Some other studies in the literature have shown some interesting results on the use of SVF. In Mikulak in 2019, treated elderly patients at 80 to 94 years, grade 2 to 4 OA. And he was able to show a quality of life improvement and a decrease in their NSAIDs and painkillers. Microfracture, better long-term outcomes when you use SVF than microfracture alone. And Zhang did a 5-year follow-up, and approximately 60% of patients at 5 years, their outcomes still persisted, and cartilage defects at 5 years were still improved and smooth. We've used, and others have used it in small joint osteoarthritis, and there's reported approximately 50% decrease in pain. Frozen shoulders and rotator cuff tears respond very well to SVF treatment. And some patients treated approximately out 4 years on avascular necrosis of the hips, AVN. We've seen a stabilisation of the disease, and we're now doing some ongoing studies on this to see what results that we could obtain. We're beginning some studies, and we've done some studies in the States on chronic low back pain, and we would see this area of research is beginning to gain entrance in improving degenerative disc disease. In patients, we've had some very good responses in our patient in the States, and we intend to look at a more formal study here back in Australia. And the reason why this is of interest, I suppose, in Australia is our orthopedics and neurosurgeons cost is generally around $50,000 to $70,000 when they go in for surgery, why our treatments is a lot lower in cost. So some of the early studies here are showing a decrease of 50% over 2 years, but we have to be careful when we look at these that disc problem is generally a joint issue, degenerative disc, and an irritated nerve root. So we have to treat all of those problems if we're going to get better results than this. So currently there's approximately 7 regenerative cell studies looking at degenerative disc disease. I'd just quickly like to talk about the mechanism of action in orthopedics, because it's one of the main questions we're asked. Osteoarthritis is a progressive loss of joint function, and the inflammatory mechanisms appear to be the chief agents now for structural degeneration. So it's now thought that there's an imbalance between the anabolic, the cartilage forming, and the catabolic cartilage destroying processes driven by the synovial inflammation. So persistent synovial inflammation leads to cartilage destruction. So there's been a very good study by Lepente in 2020 when he injected the SVF and followed at 12 months, looked at the synovial fluid pre and post in patients. And there was a decrease in the catabolic and pro-inflammatory molecules MM2, interleukin beta, IL-6 and IL-8, and an increase in the anabolic and anti-inflammatory molecules IGF-1 and IL-10. So there's direct evidence in patients of immunomodulation by the SVF for knee osteoarthritis. So I suppose we've now we like to use stromal cell therapy to address the gap between standard analgesics and surgical management in the use of knee osteoarthritis. Our data indicates approximately 94% of patients respond to the treatment, and we've treated quite a few thousands of patients. There's evidence of an average of 75 to 84% improvement with a single injection in the knee. For the hip, that's not so good, and we see only approximately 65% improvement. And most patients return to a normal level of activity, and there's a decrease or ending of use of inflammatories, and we see in the associated side effects. And it's very important to monitor this decrease in their painkillers. And we see improved activities likely to result in a reduction in other comorbidities associated with osteoarthritis. So that's basically my talk. And happy to take any questions.
Thank you, Doctor. Dr. Ooi, would you like to take over?
Yeah, I will take over. I will say thank you to Dr. Wing. Actually, we still have a few minutes. We can have a few questions for all of you. Dr. Chung, are you there? Dr. Chung? Oh, I think she's not responsive. By the way, Dr. Wing, I saw your slide. Your products in Australia, Taiwan, India, US, not in Malaysia.
No, we're not in Malaysia. We'd like to come to Malaysia. Yeah.
Oh, all the Malaysia on the line, they heard for this. I think not only for you, also the Taiwanese products. Today they will share a lot of Taiwanese experiences and products. I want to ask Dr. Cyrus, are you there? Dr. Cyrus? Dr. Yang?
Yep, I'm still here.
Yeah, Dr. Yang, yeah. I really enjoy your talks about the many design, many regulatory designs. I think just like Dr. Wayne said, many regulatory depends on different countries. I wonder whether you have experiences the difference in between the Taiwan or other countries. Of course, maybe the Malaysia too. I did not know, but I want to ask you any experiences for that to remind all of us.
Yeah, sure. I think Taiwan is following sort of like more like a PIC GMP regulation. So it's more in line with the EU regulation in terms of its manufacturing. US, they have a GMP system, which is slightly different, but highly similar. So generally, once you get through your manufacturing process, like your CMC, get approved by one of the regulatory agency in either US or in EU, it's a good ticket to enter another market. In Taiwan, if we look beyond manufacturing, we have the Regenerative Act draft in our legislature, and I think with this, once it get passed by our legislators, we will have the advantage to bring our cell therapies to market after phase 2 with conditional approval, with a 5-year conditional approval. So this is a huge boost for the Taiwan cell therapy industry. So the regulations slightly complicated, but the general idea is that they want to grant faster access for all the product to the patients in Taiwan. So we also believe that this make Taiwan a very good market for other countries like Malaysia or other countries in Southeast Asia, because a biotech can bring their product in Taiwan past phase 2 and have early market access. So I think that's the advantage of this market.
Yeah. Thank you. Your reminder that actually for my information, Malaysia, I think just this year or last year only, they are starting the regulation of stem cell program. Actually, many also we can learn a lot from the Taiwan or Australia or US or other country. Actually, Southeast Asia is a very huge, a very big country or big market or big population or big opportunity for you all. I think Malaysia is one of the center of the heart of Southeast Asia, and then we can cooperate with them. But before I end this seminar, I want to ask you again, all the doctor, because Dr. Chung, are you there? I want to ask Dr. Chung.
Yes,
Dr. Chung.
Yes.
Yes,
Tony Chou, yes.
Yeah. Sorry, I want to ask you because I have a few patients. Even he was a doctor, spinal cord injury, bedridden, many times.
Traffic accident.
Yeah. So, how do you manage this neurogenesis or anything?
Yeah. So I think nowadays we still have some neural rehabilitation for the spinal cord injury patients. But then I think this topic about the stem cell therapy, that's the new hope for these patients with the paraplegia because of the spinal cord injury. So in my presentation, we have the first patient in Taiwan last year. The patient had the traffic accident and the weakness and the paraplegia of the lower limbs, and he received the injection of the autologous bone marrow mesenchymal stem cell injection at the spinal cord injury, the damaged site. And I think that's last November in Hualien, the Niann-Tzyy Dai hospital, they will follow up this case. And as far as I know, they also apply the spinal cord stimulation to this patient. So not only the stem cell transplantation or injection, if we want to promote or facilitate the neuronal conduction and then the function, maybe some physical or physics like the spinal cord stimulation, like the electronic or the magnetic stimulation to promote the function of the stem cells. Yeah, so there's a combination therapy. So not only the neural rehabilitation therapy for most patients with spinal cord injury. Today, this year, we have the patient, they can receive the injection of the stem cells. And then the combination therapy, they combine with the spinal cord injury with some physical, like the magnetic or electric therapy to have the best performance of these treatments.
Yeah, thank you. I asked you this one because most of spinal cord injury patients, they are younger.
Yeah,
so I think so all of doctors here, you are doing the cell therapy. I think we should focus on this type of patient because they are still younger. They want to move up. They want to make an action. but they very disappointed. I think maybe in the future we can continue to have this
that's a challenge.
advanced systems. Yeah, it's very challenging and also a very new hope for the patient and for you guys today on this seminar. Am I correct?
Yes, absolutely.
Yes.
Dr. Hwang, may I have one more question?
Yes, sir.
Thank you.
Go on.
I just heard from Dr. Wayne's lecture. It's really a wonderful lecture. I've contacted with StroMed technology for several years, and we've used it to treat some degenerative disease and using this technology. As I know that this StroMed SVF technology is a kind of cell isolation technology. So we can just apply this technology for a clinical application. And I heard from Dr. Wayne, he used a lot and got many good response in the orthopedic treatment. I would like to ask Dr. Thomas about what about for the systemic application of the SVF, especially from the StroMed technology. How do you think about the future and the potential for this application? Thank you so much.
Thank you. I think there's very good applications using it systemically. Anecdotally, we've seen some very unusual results, improvements in cardiac. We've seen it with migraines improvement. So it really does travel through what we see, and there's been some studies which show it hones to areas of inflammation. So I think there's going to be some very good uses for it, different applications with different diseases. I suppose it just needs more work, more studies done on the applications, because these cells do hone to areas of inflammation. So it's some of these patients, it does surprise you. They come back and you think, they see improvements in other areas of their chronic diseases.
Thank you so much.
Okay, thank you. I'm wondering, Dr. Wayne, in your slide, you say your product is low cost. What you compare to low cost? Because many doctors here, their slide never put the very low cost. What you compare to? Compare to the surgeon or compare to other?
Yeah, look, I think low cost compared to other surgeries, low cost, I suppose, in the consumables, and really the cost is the doctor. The cost is the doctor. It's not really our. So we leave that up to the doctor, not to ourselves. We just provide the technology.
Yeah, it's actually for even the patient's family. Sometimes they ask me, just like Dr. Dai. They say my grandpa got a pressure sore. I had to spend such a lot of money to carry the pressure sore. And what caused the pressure sore? The grandpa is a long-term bedridden.
Yeah.
The cost and the profit in the family.
Look, our technology is low cost. It's really about how it moves forward in each country and the doctor's charges. Yeah.
So I think today, thank you all of you. I think I'm very enjoying your knowledge sharing. Actually, your knowledge sharing is a very good opportunity for Malaysia, learning from all you guys from Taiwan and Australia, especially because the time is very limited and almost is 5 p.m. in Taiwan. So I think that's I will end this seminar.